Hello, everybody. This is the next part of our lecture for week 7, and I did want to rewind just a smidge about thinking about oxygen usage and tolerance patterns in microbes. So essentially what we have are those organisms that love oxygen, okay? They are thriving in normal atmospheric concentrations of oxygen. So we think about those as the aerobes. Now, if you are obligated to live in those environments where you cannot survive, then we call you an obligate aerobe. So that was our first example there. So the microaerophiles mean essentially the normal concentration of atmospheric oxygen would be harmful to them, but they can tolerate a small amount and need a small amount for their metabolism. Okay, so that kind of makes sense. Microaerophiles means they need a small amount of oxygen. So when we move on, the anaerobes are on the other end of the spectrum. So they lack the metabolic enzyme systems to even deal with oxygen. So essentially, they thrive in low oxygen environments or no oxygen environments. So that would mean completely anaerobic or without oxygen. So when you think about obligate anaerobes, they cannot tolerate oxygen, period, the end. But we know that there are some things that can tolerate a small amount of oxygen. So the aerotolerant anaerobes do not utilize oxygen but can survive if it is present in their atmosphere. So these organisms can tolerate a little oxygen, but in general, they thrive in low oxygen environments. So you can see their examples are some lactobacilli. So when we eat yogurt, we are ingesting lactobacillus, and these take up resident in our large intestine, streptococcus. So we have streptococcus in our throat, and clostridial species. So thinking about Clostridium difficile being in the large intestine. Now the streptococci that would be these aerotolerant anaerobes are not likely to be in the throat. They're likely to be somewhere else in the body where there's just not that much oxygen around. Okay. Then we have our facultative anaerobes. So when you see facultative, I think optional. So these do not require oxygen, but can use it when it's present. So it's kind of like, my husband's a flexitarian, so he's not a vegetarian. I'm a vegetarian, so he's a facultative omnivore, I guess, or carnivore, because if there's meat present in a family function, he eats it. So facultative means that you can be flexible and you can use it when it's present, but generally speaking, you're more, for this particular example, you're more thriving in low oxygen environments. So these are many of our intestinal bacteria, and then also some staphylococcus or staphylococci. And this figure comes straight from your book, just summarizing the patterns of oxygen usage in various types of bacteria. Okay, so if we can imagine that bacteria have different preferences for oxygen, we can also imagine that there are bacteria that have evolved for different pH conditions. So we're normally thinking about pH of 7 as being neutral, and below that, we are in an acidic range. So low pH means acidic, high pH means basic. If you're highly acidic, then you're usually down on that 0 to 1 to 2 end. And if you're highly basic, then you would be on that upper end, 12, 13, 14. So here we see some examples of some acidophiles. Phile comes from philic, which means loving. So these organisms love the acidic environment. So we have Euglena, which is one species of, essentially we would call them protozoa. They have chloroplasts like algae and like green algae and our plants, but then they also have functions or features that make them animal-like. So they have flagella, and they also like to ingest or phagocytize food. So Euglena Eutabilis grows in acid pools with a very low pH. Thermoplasma also lives in low pH. Here they're referencing coal piles, and Picrophilus is another one, low pH. And then it also mentions here on this slide and in the book, many molds and yeast tolerate acid. So this is why in something like pickles, I recently ate some pickles and I kept thinking, well, they don't taste bad, but they don't taste like normal pickles. So likely there was a food contamination in those pickles. So essentially, we know that yeast and molds can tolerate acid, so therefore could colonize and spoil food that has been pickled. So then on the other end of the extreme, the alkalinophiles. So alkaline is that basic range or the higher range of our pH spectrum. And so they like, essentially those situations. So oh gosh, I haven't even said this before. Natrianemonas live in hot pools and soils up to pH 12. And then Proteus, which is kind of interesting, Proteus can essentially colonize the urinary tract and essentially convert that from more of an acidic, slightly acidic to slightly basic. And so essentially making that urine more neutral, and essentially helps to create an environment where it thrives. So essentially it's raising the pH from the slightly acidic to slightly basic so that it is in fact thriving in that environment. Pretty interesting. All right, so then we go to osmotic pressure. So you might remember we were talking about osmosis and diffusion. So essentially when we're thinking about osmophiles, these are the organisms that can live in high concentrations of solute. And the one that I want to talk about is the halophiles. You might have heard of the Dead Sea, which is highly salty, has a high concentration of salt. So we know that there are particular microorganisms, especially bacteria, that can grow in those high salt environments. So you start to think, okay, if my cell is tolerating all this extra solute, then my cell must have some way to compensate for that high salt. So normally you would expect that water to move from inside the cell to that outside environment, trying to equalize that osmotic pressure or the concentration of water. And so they have other ways to deal with that. So again, we have our obligate halophiles. That's where they grow. They need this higher than what you'd expect normal concentrations of salt. We're usually thinking, at least in humans, 0.9% or normal saline is kind of that ideal salt concentration in our cells. And so when we give people fluids, we want to keep with that concentration, unless, of course, we're trying to move fluids into the cells or outside of the cells. Next, facultative halophiles would be like, all right, like if salt's there, I can deal with it. But if the concentration of salt is too high, then they cannot live in that environment. For those of you in the on-campus lab, you've known already that we have a particular type of agar. And this agar is called mannitol salt agar. We're going to play with it a little bit more in the future. But mannitol salt agar favors skin bacteria because of that higher salt concentration. So it is a selective media. We are selecting for those organisms, in this case, bacteria on our skin, that will favor those organisms like Staphylococcus aureus on our skin because of that higher than normal concentration of salt. So the thing is it's inhibiting other bacteria that would not be able to tolerate that. So that's just an application of how we're going to use this information. Okay. Biofilms. So we've talked about this early in the semester. So when I think of biofilms, I often think of an analogy in ecology, where when an area which has been previously uninhabited by life becomes inhabited by life. So an example, when a glacier melts and retreats, it exposes bare rock. So in the situation of this is called primary succession. So it's where organisms are colonizing a substrate or an area which has not been colonized by life before. So the first things that move in are bacteria, and the bacteria have some acids that start breaking down that rock and just having small amounts of essentially nutrients for other organisms. The next thing that moves in are lichens, and lichens do the same thing. We know we've seen various type of crustose lichens or foliose lichens growing on sidewalks on campus, and essentially those lichens are also releasing acids which are starting to make that environment actually more favorable for small flowering plants and mosses and so on. So biofilms are not that different. So you have these pioneer species or the pioneer bacteria that move in, they colonize the surface, they begin secreting these what we call extracellular. So they're like releasing chemicals to the outside of the environment that literally helps them stay on the surface that they're trying to grow on, and essentially is going to be a source of attachment for other colonizers. So these are the other bacteria that are going to move in and become part of this biofilm. You can see in many, but not all biofilms, other species join and may contribute to this extracellular matrix and even participate in essentially foraging and sensing environments for them to move into. And then lastly, we get really some accumulation of other kinds of organisms building part of it. And you can see in step 4, biofilms serve as a constant source of bacteria that can escape and become free living. So you have this community of microorganisms, and some of them can break off and then colonize new surfaces. But when they're in the biofilm, they're actually somewhat protected from immune systems, from breakdown from acids, and so on. So biofilms are very incredibly interesting and create a big problem when we're thinking about those surfaces in the medical field that can become colonized. So thinking about catheters, thinking about not really IVs necessarily, but anything that's going to be put into the body from surgery or any instruments or things like that would have to be completely sterilized. Okay, this is the end of week 7. Let me know your questions. Have a good day, y'all. Bye.